Hi, good morning, everyone. So quick reminder that this week is an article summary share week. So tonight you will turn in, or by tonight, you'll turn in your article summary assignment. And then on Friday, we'll be meeting in our separate sections, section 1 and section 2, to do our article sharing. Okay. For those of you in section 2, I'll send you an email today reminding you or giving you some suggestions about where I would like you to be now in terms of making progress with putting together references or citations for your paper. Okay, and then we will talk more about that on Friday when I meet with section 2. Okay. Let's continue talking about other very notable genes and proteins that are real standout examples of things that help regulate the process of transformation and progression to cancer. And so one of the most important or high profile proteins involved in cancer is p53. So I think many of you are already familiar, at least in passing, with p53. Maybe you've seen it come up in some of the articles that you've been looking up and reading. So let's talk about p53 and how p53 works, what its purpose is, and how it is controlled by other proteins such as Mdm2. All right. So this is something that we looked at last time as kind of a summary slide. And we said that this helps us appreciate that at any given time, the cell, especially in, let's say, G1 phase of the cell cycle, right, is listening for different types of messages coming in from the outside onto its cell membrane and being transduced into intracellular cytoplasmic signaling pathways that are trying to nudge the cell to do one thing or the other thing. And in terms of cancer, that one thing versus the other thing is, should I stay in G1 and keep doing my normal differentiated cell functions? Or should I give up on that, right? Put that on hold and focus on moving forward into S phase and eventually to mitosis and dividing. Right? And we said that these factors are governed by many overlapping competing pathways. The oncogenic pathways are the ones that are in orange, right? And the braking pathways, in other words, the tumor suppressor pathways, are the ones in blue. And remember, we said last time that ultimately all of these sort of like layers of brakes, acceleration, they all end up kind of focused on the function of the RB protein. Right? And importantly, the phosphorylation status of RB that will then determine whether the cell cycle plunges forward over the restriction point or the R point in late G1, after which the cell no longer listens to any changes to external messages. It's on this autopilot program headed towards mitosis. So when we think about the cell then as a two-footed driver, right, constant, some pushing of the acceleration, some pushing of the brake, and which one wins out, right? As the cell goes through the cell cycle, if it goes past the R point, then in reality, right, it doesn't listen anymore to signals, and so it's committed to getting all the way through. But being committed to getting all the way through to mitosis and dividing into two daughter cells does not mean that there are not additional opportunities along the way to make sure things are going okay. If problems arise, right, they might still be repairable as the cell moves from S phase to G2 and even into mitosis. If they are not repairable, maybe you don't have many options. So let's say the cell moves past the restriction point, so it kind of moves into S phase, it duplicates its genome, and then at the end of S phase, it kind of detects, whoa, there's a problem here, right? There's a problem that happened with the maybe the replication of the genomic material. So some problems, like minor DNA mistakes in replication might be repairable by just some enzymes going to work and fixing something, right? But bigger problems that are part of improper genomic replication, for example, if you have translocation events that happened, or aneuploidy, right? For some reason, like some of the chromosomes got replicated, but some of them didn't. Deletions happened. Maybe a whole chunk of some chromosome just disappeared, right? Or even things like chromosomal segregation as you're moving into things like mitosis, right? If the sister chromatids don't actually separate appropriately to go into the daughter cells, if things start moving in one direction or another direction improperly, those are pretty gross macro scale problems that the cell may not be able to fix, right? So if the cell encounters that, what is it going to do? Well, it's certainly not going to back up and go back into G1 phase, right? Because remember I told you, it's not like the cell can be like, hey, I tried this, this just isn't for me. I'm going to try this again some other day. I just want to go back to G1, right? And be a mucus producing gland cell, right? No, at this point, the cell is committed to finishing this through, but if it cannot finish it, what choice does it have, right? Well, sadly, there's not much of a choice. It might need to choose the nuclear option, right? It's got to push the button that's, I don't know why they always look like this, and that leads to apoptosis. Right? So apoptosis is like the nuclear option, right? So what is apoptosis? We usually think of apoptosis as the cell just kind of destroying itself. So we think of it as a bad thing, right? Oh, that's so sad, right? You had no other option, but you had to actually destroy yourself and take yourself out of the picture. But the reality is, right, that apoptosis can be thought of as friend of the healthy and enemy of cancer. Right? I kind of think this is like a movie poster, right? That's why it's kind of set up this way. So it's all dramatic. Right? But what do I mean by the friend of healthy an enemy of cancer? Because, right, the better your cells are at being able to detect that there's something grossly wrong with their genomes, maybe due to some sort of mutation that led to a translocation or something like that, and the better they are at just pushing the button and taking themselves out of the picture, right, they do so to actually spare you as the whole organism. That is kind of like a neat thought, right? Because we think of individual cells as being very adaptable, and over eons of evolution, cells have different mechanisms for dealing with things like changes in their environment, like rising, falling oxygen levels, nutrient depletion, things like that, right? So cells are sort of pre-programmed to do whatever they can to survive. And yet, right, when things get really bad and they have issues with their genome and the integrity of their genome, they also have to make that decision that, you know what, even though I'm geared up for survival, at this point, it's better for the whole organism, right, if I just go ahead and undergo apoptosis, right? It's an interesting way of thinking about it. So that apoptosis, right, at least intrinsic apoptosis caused by a cell detecting that there are these problems with its genome, right, is quite a sacrifice. Right? The cell is sacrificing itself to spare you as a whole so you can continue to live free of the consequences of its genome being messed up. Right? So that's why we say it's actually the friend of the healthy and enemy of cancer. Right? So the better cells are at undergoing apoptosis, right, the less likely they will become cancer. So let's explore some of these underlying factors that govern this sacrifice decision that the cell has to make. Some early experiments back in the 70s, late 70s, were trying to identify different types of oncoproteins, right, that were involved in driving transformation forward. So back then, 60s and 70s, everybody was about oncogenes and oncoproteins, right? Because they were really enamored with this idea that it's, mutations in some proto-oncogene like RAS or SARC, right, turns them into this totally evil oncoprotein, right? So as people were researching it, some groups were looking at things like the SV40 large T antigen, which we already learned is one of these proteins made by certain tumor-causing viral genomes that contribute to the transformation of cells. So in this experiment, they were looking at the immunoreactivity of SV40 transformed cells. So in this experiment, they would do things like, for example, if you look here, they would take totally normal, healthy mouse cells, like mouse fibroblast cells, and then some of them they would leave alone, right, as a control group. So this is whether they get transformed or not by the introduction of SV40 virus. So you have a group that's the control, no. The other group, you go ahead and you transform, right? You expose them to SV40 virus that ends up causing them to transform. And then what you do is you lyse these cells to take out all of the proteins that are inside of these cells. And then you interrogate what proteins are different between the transformed cells versus the non-transformed cells, right? And the way you interrogate it is you want to say, are there certain proteins that are now appearing in the transformed cell, right, that would represent that they are transformed, right? And so one way you can interrogate that is if you have antibodies that are looking for particular types of proteins that are different in transformed or cancerous cells. So they would take the lysate from these cells, and then they would probe them with serum that comes from other animals that were already sort of exposed to either normal 3T3 cells or transformed 3T3 cells. The implication being that in those animals, right, they would build up some immune reaction to things that are different in the transformed cells versus the normal cells. So here you can see normal healthy cells that are transformed with SV40, and then you probe the lysate, all the proteins from those cells, with antibodies that came from animals that were previously exposed to cancerous cells versus exposed or animals that were not exposed to cancerous cells and wouldn't necessarily have antibodies that could detect things that are, in the transformed cells, right? And what they saw, not surprisingly, here's large T antigen, right? 94 kilodalton protein, and yay, right? So this makes sense because you transformed these cells with SV40. So now you see the SV40 large T antigen protein at its expected 94 kilodaltons, and the nice control group that was not transformed, right? Even if you probe with serum that contains presumably antibody against the large T antigen, you don't see it, right? But you do see something else, right? Look down here. Researchers were surprised to see this other 53 kilodalton protein, right? This little mark is 54 kilodaltons, but you can see the band is a little bit smaller than that, right, in the transformed cells, right? That wasn't showing up in the normal cells, right? So, is this a protein that is also from the virus, right? Or is this something from inside the cell that gets upregulated in response to being transformed? Right? What's really telling is, what if instead of normal mouse cells as your starting point, you took a cell line that's already cancerous, but importantly, cancerous not because they were transformed by virus, they were turned into cancer for some other reason, right? They got their mutations for some other reason. What if you looked at those? So these are non-viral cancer cells like this F9 cell, but you do the same experiment. You transform some of them with SV40, or they're already transformed, but you expose them to SV40, and then you have a control group that you don't expose them, and you do that same probing. And what you see is, not surprisingly, right? Right, you don't see much difference in the SV40 protein levels, okay, when you probe with these different antibodies, but you see a dramatic detection that these cells, for some reason, have much higher levels of this mysterious 53 kilodalton protein, right? And what's kind of interesting is you actually see that in the SV40 cells that were exposed to SV40 here, you actually see a little bit less of it than in the regular cancer cells that did not get additional SV40, right? So could it mean that SV40 somehow interacts with whatever this mysterious protein is, right? And what is this other protein? So now we know that, yes, large T antigen does interact with this other 53 kilodalton protein, which to this day we just call P53. Boring name, but that's what it's called, right? And here you can see, here's large T antigen. Large T antigen forms this multi-subunit pinwheel kind of a shape, or kind of looks like a snowflake. Right? And the ends of it are the right shape to bind to and sequester this mysterious 53 kilodalton protein. Right? So one large T antigen hexamer can hang on to 6 of these p53s. So what does that mean? Maybe that could mean that this large T hexamer somehow stabilizes the p53, or it could mean that maybe, it prevents the p53 from doing its normal job. So on the one case, if it helps stabilize the p53, and then you have p53 and large T antigen working together, right, in this crazy like big pinwheel shape, maybe that's super oncogenic, right? So maybe p53 is an oncogene. Or if large T antigen is in reality preventing p53 from working, right, by binding it up in this pinwheel, then maybe p53 is a tumor suppressor. Right? So early evidence definitely suggested that this p53 protein is very important and relevant to discussions of cancer. So here you see p53 point mutations are very common across a whole spectrum of different cancer types, right? Everywhere from being maybe present to only 5 to 6% of cervical cancers up to nearly 50% of ovarian cancer cells from ovarian tumors show evidence of p53 gene mutations. Right? So no doubt about it, the early researchers said, hey, we got to figure out what this p53 is. So the fundamental question, is p53 an oncogene or is it a tumor suppressor gene? Right? So this early experiment, I think is a wonderful experiment because it seems very straightforward, right? But then things kind of go off the tracks really quickly. So it's a classic experiment where they took rat embryonic fibroblasts that they transfected with expression plasmids for RAS and p53. So 2 different expression constructs, right? That when you put it in these rat fibroblasts, these rat cells make tons and tons of RAS, and they make tons and tons of p53. And then the question is, if you then go ahead and you culture these cells, would you see the telltale signs of transformation? Remember, on a dish of cells, you might have transforming cells forming little foci, right? Little islands where cells that are transformed begin piling up on each other. So when you transfected these wild type embryonic fibroblast cells with wild type RAS expression vector and wild type p53, this is what they saw. For the most part, the cells did not transform. I mean, here you have one maybe example of a transformed foci. Here's maybe another one, right? But very little activity. So they concluded, wait a minute, right? Because when you transform these cells with RAS alone, right, you should get lots of foci. But now that you have this p53 in there, you're not getting foci. So, all right, the conclusion is p53 must be a tumor suppressor, right? It's somehow, counteracting the oncogenic effects of RAS. So what they did was then said, well, okay, let's mess with the p53. In our expression construct, let's truncate the p53 gene so that it doesn't form a totally normally formed p53. What do you see? Then when you express those together, so the wild type RAS, but you're overexpressing a deletion mutant of p53, now, look, now you get lots of foci, right? Because you're like, now the RAS is working. So yeah, I guess that makes sense, because if p53 is a tumor suppressor gene, then by messing with it, by deleting part of it, and it no longer works to be a tumor suppressor gene, now you're seeing RAS working, right? So you're like, okay, that's the real conclusion, right? The p53 is a tumor suppressor gene. But the researchers took it a step further, because they knew that p53, right, is mutated in so many different clinical tumor samples, but they realized the types of mutations that you see in p53 in human cancers are not deletions. What you see are different point mutations within the p53. All right, so they said, okay, well, what if we actually just reconstructed one of these point mutations in the p53 like you would actually find in a real human cancer? Then what would the result be? if you made these cells express both. And this is where the experiment goes off the rails, right? Because this is what they got. Right? So you're like, wait a minute. So here is the wild type RAS that's overexpressed, but it's co-expressed with a p53 valine 135 mutation, right? So at amino acid position 135, there's a point mutation that puts a valine there. Right? Right? Look at what happens, right? How could it be that clearly the wild type p53 in dish C protects against RAS, right? And if you go ahead and you truncate the p53 so it doesn't work in dish A, right? It's a non-functional p53, so RAS is allowed to transform things. But if you actually reconstruct the type of mutation that you see in human tumors in p53, not a deletion, but a point mutation, you actually get increased RAS activity, right? So that's crazy. So is p53 a tumor suppressor, or do point mutations of p53 somehow make it an oncogene also? This is really curious. Another piece of the puzzle is that double knockout mutations of p53 are not embryonically lethal. So that already separates it from how other important, powerful tumor suppressor genes work. Because if you have double knockouts of TSGs like RB or PTEN, those are embryonically lethal, right? A developing embryo, whether a mouse or, presumably a human, that has lost both copies of RB or PTEN at the beginning of embryonic development, it's not going to work, right? I mean, that embryo is not going to make it to term. But yet, you could have double knockouts of p53 in mice, and they actually develop normally, right, into baby mice, and they're born. Right? But they die on the younger side. So let's look at this. Right? So p53 knockouts, or p53 wild type mice, so p53++ in this experiment, that group of mice live beyond, let's say, 500 days. If you have a heterozygous plus minus, you can see that these mice actually, for the most part, right, can still survive normally. The p53 double knockout ones, this is the surprising thing. Yeah, they are actually born, and they can survive like 100 days plus, right? But they do die earlier. Right? So clearly then, p53 is not necessary for normal cell division at the embryonic stage, right? So it's not like RB or PTEN, something like that, right? So that's why people said, oh no, so forget it. It's not an important tumor suppressor gene, right? Or is it? Right? So I mentioned to you that the curious thing about p53 mutations in clinical samples is that the majority of mutations that happen in tumor suppressor genes that lead to advanced tumors, as we've already studied, usually have to do with parts of the tumor suppressor gene being deleted. Right? So by far, in things like APC, right? Remember that help regulate beta-catenin, or other tumor suppressors like ATM or BRCA1, right? Half or over half of the types of mutations that you see in human clinical biopsies, right, involve deletions, right? So that's caused by frame shifts that result in, a premature stop codon, right? So therefore, the gene or the protein product, not all of it gets made. Right? Relatively few of them in APC are due to things like missense mutations. Right? Remember, missense is when you substitute one amino acid for the other. Missense mutations are a bit more common in ATM and BRCA1, but clearly not as important as frameshift mutations leading to truncations. But look at p53. Right? Overwhelmingly, the type of mutations that you see in clinical samples are point mutations, right? Missense mutations that result in an amino acid somewhere in the p53 changing from something to valine, right? For example, that was the example used in that classic experiment before. So how do we make sense of this? Right? How could it be that p53 behaves so differently than other tumor suppressor genes in terms of the nature of mutations? So if we look at the functional domains of the p53 protein. So here it is. Here's the N-terminal end of the protein, the peptide, right? And here's the carboxyl terminal N, so the N-terminal, C-terminal. The whole p53 protein is 393 amino acids, and it can be divided into these different functional regions. This purple region here is what we call the transactivation region. So in other words, people have now understood that this is the region that determines whether p53 is told to go ahead and function. We have proline-rich regions here, which, proline-rich, we've talked about that before. What's the significance of proline-rich regions in proteins? Oh, oftentimes that helps other proteins that have domains like SH2 to bind to these proteins and help organize these proteins, right? Bring other proteins together so that they can work together. There is this region over here, this peach-colored one, which is the tetramerization domain, right? Which means single p53 molecules have the ability to partner up with 3 other p53 molecules to form a tetramer, right? And this is really the key, right? Because there's this tetrameric domain, but this middle part probably is the most relevant to our discussion about point mutations. This is the DNA binding domain, right? So this blue-green region here, that would be this region here, right? That actually binds to the major and minor grooves of DNA. What are these numbers? 175, 245, 248. These are the amino acid positions that you most commonly find point mutations in clinical samples of tumors that have p53 mutations, right? And notice they're not in this purple region. They're not in this tetrameric region. They're in the DNA binding region, right? That's kind of mysterious, right? This is the reason why this works. So here you have a tetramer of p53, 1, 2, 3, 4 molecules of p53 bound together by their tetrameric domain, okay? So these are these domains that are helping them form a tetramer, and they're interacting with DNA through their DNA binding domain. Importantly, all 4 of these need to be able to have a functional DNA binding domain in order for them to contribute to the whole tetramer being stable on the DNA. If any of these had a compromised DNA binding domain and could not bind to the DNA, but yet could still bind as part of a tetramer, it would just end up messing up the ability of the tetramer to actually associate with DNA. This is why you have this very strange sort of phenomenon with p53. Because as I pointed out to you before, p53 plus minus genotypes are dominant negative, right? Meaning that even though in other types of tumor suppressor genes like P10 or RB, if you're RB plus minus or P10 plus minus, you're still okay in the sense that that plus allele means you're making the functional form of the protein, so you have some of it to do its job. Why is it that P53 is different, right? In P53, if you're plus minus, you end up having those baby mice, right, die a bit earlier, okay? It's because of the fact that p53 functions as a tetramer. So imagine this, right? In a cell whose genome is p53 plus minus, in other words, one copy, maybe the one that came from mom, is wild type, where the other copy, the one that came from dad, right, either got a hit because it got mutated, so there's a point mutation in it, or maybe, allelically, maybe there was some allele that it got, right, that doesn't function right. That means the p53 proteins that are made by that particular cell would be a mix, right? The blue ones represent the wild type p53 that's being made from mom's good copy of the gene, right? The red one is the defective or point mutant p53 that's made from dad's copy of the gene. When these blue and red p53s are floating on the cell, they will come together to form tetramers, right? But as they form tetramers, they can be in any combination. So that means only 1 out of 16 possible combinations of a p53 plus minus cell would be the normal wild type one, right? Because in only 1 out of 16 cases would all 4 subunits be the blue wild type p53, right? You would have 1 out of 16 that all 4 of them are the, all 4 of them are the mutant form, but everything in between, the other 14, right, are a mix of red and blue. And as I said before, the moment you have one of these units that make up the tetramer having a defective DNA binding domain, that messes up the ability of the whole tetramer to stay on the DNA. So let's look at this. If you have a wild type embryonic stem cell that's p53++, right, wild type, and let's say you get some sort of deletion event so that you completely mess up one copy of the p53. So this would be like resulting in a truncation, right? So you still make the wild type form of p53 from one of the alleles, but the other one now doesn't make p53 at all, right? Because the form of p53 that it makes maybe is so truncated that the p53 is not even kept in the cell, it's degraded. So in this case, right, the function is almost normal because all you have is wild type p53 in there. But if the nature of the mutation is not a deletion, but a point mutation, right? So that's typically what we talk about when we say p53 plus minus, because remember the clinical samples show that it's really point mutations that you see. Then you have one copy of the good, but one copy of the point mutant. So this is basically one blue subunit, one red subunit, and p53 function, right, is almost completely impaired. Right, so loss of p53. So how does that fit in with our discussion about apoptosis? We said that if the cell is committed to going past the R point and going through S phase, G2, and mitosis, if there are different problems that it encounters along the way that it cannot cope with and fix, these could be problems that are wide-ranging. For example, suddenly the cell doesn't have enough nucleotides to even replicate its DNA, replicate the genome, right? So a lack of nucleotides, what's the cell going to do, right? If it doesn't have enough A's, T's, C's, or G's to finish replicating the genome, well, that's bad, right? Or radiation. What if the cell gets hit by radiation that starts actually messing up parts of the genome as the genome is being replicated, right? That's really bad too, right? Because what are you supposed to be copying now if things are all damaged? If, for example, the cell during G1, right, gets way too much oncogenic signals, right? Oncogenic signals that are just forcing the cell through the cell cycle into S phase, even though the cell wasn't ready to go into S phase, right? So it almost got plunged over the restriction point before it had a chance to properly build up proteins that are needed for S phase. That would be really bad. Right? Changes in metabolic status, like if suddenly it's a hypoxic condition and the cell cannot do its normal cellular respiration, right? Then maybe it shouldn't bother finishing the cell cycle, and other things that might just block transcription, right? So as genes, necessary genes for completing mitosis are being transcribed, what if something gets in the way? So in those cases, maybe the cell simply cannot deal and it has to push the button, right, and undergo apoptosis. So p53 is the button, right? So p53 then, you suddenly realize, oh, it actually is a very important tumor suppressor gene because it makes the decision whether to allow the cell to continue on with the cell cycle. And so p53 can control things like cell cycle arrest, or maybe, send the cell into senescence, G0. It will also decide whether things are okay to return to proliferation. p53 is involved in things like DNA repair, so sensing damaged DNA so that it can halt the cell cycle, so the cell might have a chance or shot at repairing the DNA. p53 can also prevent angiogenesis. In other words, angiogenesis is the growth of blood vessels heading towards the cell. And ultimately, p53 can trigger the self-destruct program, right, which is apoptosis. You know, when I show the little nuclear button here, I always think about in movies, right? In movies, it's like when the spaceship that everybody's on, they need to like blow it up or something. Everybody evacuates except for like the captain, because the captain heroically, she or he stays behind to like push this button, right? And then when they push the button, this like computerized voice starts counting down, right? They have 60 seconds to get to one of the escape pods or something like that, right? And I always thought that was really weird, because I was like, that's kind of dumb, right? Because wouldn't it be better if you just put this button inside the escape pod? That way you can get there first and then blow the ship up, right? But anyhow, the point is, right, just like in the movies, right, the self-destruct sequence is a carefully orchestrated set of actions that will end up with the spaceship blowing itself up, right? Apoptosis is like that as well. So we will take a closer look of apoptosis in a subsequent lecture, right? But p53 controls that, along with these other decisions about whether the cell should pause, try to do repairs, and if everything looks okay again, be free to resume the cell cycle. So how does p53 do its job? p53 works through different intermediate proteins in order to do things like halt the cell cycle. We already looked at some examples of tumor suppressor genes whose job it is to put the brakes on the cell cycle. Right? We looked at things like, for example, p21. Right? p21 is one of these proteins that interacts and prevents some of the cyclin CDKs from doing their function. Oh, so not surprisingly then, the activation of p21 to get it to start putting the brakes on is controlled by p53. Right, so you can see here, this is a group of cells that were wild type p53++, right? And they were exposed to radiation, right? Strong doses of X-ray radiation that caused just dramatic damage in many different parts of the genome inside of the cells, right? So in other words, the damage is so extensive that the cell cannot repair the extensive damage, right? And as a result, what does the normal cell, the normal well-behaved cell, need to do? If it cannot repair the stuff, it needs to push the button, right? So if it's wild type p53, this is what you see. Time in hours following the exposure of this high dose of X-rays, and then on the y-axis, you're measuring the percent of cells that are still living after said time out to 25 hours. And what you see is that the wild type p53++ cells, right, they do the right thing, right? They undergo apoptosis. So by 25 hours out, you're down to about 20% of the cells are still alive. But if you have cells that are p53 heterozygous or p53 knockout, what do you see, right? They don't do the right thing as readily, right? Especially the ones that don't have p53 in them. They continue surviving, right? And that's really scary. Why? Because they're surviving with their grossly messed up genomes. Right? That's why you go to the dentist, right? And they're going to take some X-rays, right? And so they always put that lead apron on you so that they're only zapping your jaw, right? So think about this, that level of X-rays, of course, is not as dramatic as the level of X-rays in this experiment here, but right, those X-rays are causing DNA damage. So that's why, right, since you have to take an X-ray of your jaw, you have no choice but to leave your sort of face exposed. But that's why you might as well cover up with a lead shield to protect the other cells of your body from being exposed to the X-rays. Because even though you hope, right, that since all of your cells have p53 in them, and wild type p53, that any cell that does get damaged by the X-rays, for example, in your jaws, right, you count on the fact that those cells, if they're too messed up, will just undergo apoptosis, right? So you don't think about that, right? That after getting a jaw X-ray a few days later, you've probably lost several 100 million cells from your face because they underwent apoptosis. But that's okay, because those cells did the right thing and they're constantly replenished by new healthy cells, right? You worry that some of those cells, right, might get mutations such that the p53 isn't working properly. Because what if, for example, the parts that get messed up in the genome by chance involve the p53 gene in some of those cells, right? That's not good. So that's why X-rays do cause cancer, right? And that's why you protect your body. So at least it's just the cells in your jaw that are exposed to your dental X-ray and not the rest of you. Okay. How is p53 regulated? So very clearly, our cell must be ready to use p53 at a moment's notice, right? Because if your cell encounters something horribly wrong with its genome, it can't wait 6, 8, 10 hours to be like, oh yeah, we should start turning on expression of p53, right? So let's start doing that. No, right? Because other horrible things might be happening in the meantime, like more mutations, right? So here we see p53, it's expressed, again, it comes together, 4 units come together as a tetramer, right? But normally, p53 shouldn't be functioning, right? Because you don't want p53 always putting the brakes on your cell cycle, right? Because that would kind of mess things up. So this is one of those situations where your cell wants to have p53 ready, but it doesn't want p53 to work, right? So again, the way that your cell deals with this is it's constantly making p53, but at the same time, rapidly turning the p53 over and degrading it. So here you see, here's a p53 tetramer. If it were allowed to work, it would go ahead and go into your cells, into the nucleus, and start binding to certain genes that would start the brakes, right? So things like p21, things like that. Right? So the way that our cells prevent that from happening is almost right away, p53 gets acted upon by this other protein, Mdm2. And Mdm2 is an enzyme that transfers ubiquitin groups. Ubiquitin groups are these small little amino acid sequences, right, that serve as little flags when they are added to other target proteins to post-translationally modify them. So Mdm2 is what we call an E3 ligase. Right? So in other words, it ligates or transfers ubiquitin onto a target. Once Mdm2 ubiquitinates or ubiquitinates the p53, that is a sign for the p53 to get fed into the proteasome, the recycling bin of the cells, and the p53 is just degraded, right? So this is kind of like the concept of how beta-catenin is regulated, right? Our cells always make beta-catenin, but beta-catenin gets phosphorylated by the APC-GSK3 complex, and then that phosphorylated beta-catenin gets sent for recycling, right? Here, it's p53 being targeted by Mdm2, and it gets ubiquitinated, and that sends p53 to the bin. Notice, this is kind of interesting here. p53, if it's allowed to work, one of the genes that it controls and turns on is actually the Mdm2. Right? So with increased p53 activity, you get more Mdm2 expression, right? So here's the mRNA from the Mdm2 that gets made into more functional Mdm2 proteins that feed back, right, and actually inhibit the p53. Right? So you can get the sense that the p53 is very conservatively regulated, right? If p53 is allowed to work, one of its products, talking to different genes, is the very protein that will then quickly eliminate the p53 itself. So the normal half-life of p53 in our cells, a p53 molecule, is less than 20 minutes before it gets targeted and degraded. So why is this a good control system? It's a good control system because the moment a cell detects that there's something horribly wrong with its genome, right, it can just very quickly deal with making use of the p53 that's already there by preventing the p53 from being associated with the Mdm2, right? So all you have to do is very quickly block the association between Mdm2 and p53, and then the p53 will be allowed to escape from being ubiquitinated and degraded, right? So that works. So that way you can have a large pool of p53 sort of building up all the time right? And the moment you need to keep that pool, you just interfere with the MDM2 binding to the p53. So how does that work? Remember in the protein map that I showed you of p53, we said that here's this tetramerization domain, so it can form 4 subunits of p53 come together. There's the DNA binding domain, right, where we see most of the most important clinical point mutations in aggressive tumors are in this region because it messes up the way that the tetramer can then associate with DNA. And I mentioned there was that purple transactivation domain. So let's look at what this is about. I said this is kind of like an on and off switch, not really on and off switch, but this is how the p53 gets activated. p53 can be phosphorylated, right, in this purple transactivation domain. Right? Why is that important? Look at this really cool figure up here, right? So this is 2 proteins pressed up against each other. The green tennis ball ones are the electron orbitals of the p53. Right, so that's the 3-dimensional shape of the p53 protein. And then the MDM2, right, is shown in this kind of like transparent net so that you can see through it. And you can see how perfectly the p53 fits with MDM2. And that's why if this binding happens, MDM2 can then easily start adding ubiquitin molecules to the p53, right? But if you phosphorylate some of these p53 residues that are in this transactivation domain, right, that's the surface here where they're contacting, you mess up the ability of MDM2 to bind to the p53, right? If the p53 is a different shape here due to phosphate groups that are hanging on there. So that's how it's done. When the cell needs to get p53 to work, you just have to phosphorylate it in this domain, right? So this is the last slide for today. So that means here we have the p53. So this builds on the cartoon that we looked at in the previous slide. The p53 normally gets acted upon by MDM2, right? And the MDM2, right? This is a little confusing, but in order for the MDM2 to work, it itself gets phosphorylated just to activate it. Okay, so the activated or phosphorylated MDM2 is able to bind to the p53. That results in the p53 being ubiquitinated and degraded. Right? If, however, there is DNA damage in the cell, there are proteins that can detect damaged DNA. We will learn about these in a subsequent lecture. But the point is, they signal a family of kinases, the checkpoint kinases, like checkpoint 1, 2 here. And these are the things that perform their kinase function on p53, right? So they will transfer phosphate groups into that transactivation domain of p53 so that the orange MDM2 no longer binds to it. And then this phosphorylated p53 then goes to work, turning on other genes, right? And I did mention that one of those genes is the MDM2 gene itself, right? So it's kind of ironic in a way that the activated p53 binds to MDM2, right, and makes more MDM2 that will then eliminate or reduce the function of the p53 in this negative feedback loop. Right? And this is kind of neat because that means the more you can encourage MDM2 production, the less you're able to have p53 functioning, and the more easily the cell plunges forward through the cell cycle, even if it shouldn't, right? And so that's why, look at this. Here, another thing that kind of intersects with MDM2 regulation are mitogenic and cell survival signals, right, that are coming from the outside of the cell. Right, so these strong mitogenic signals also encourage lots of production of MDM2, right, to keep p53 function suppressed. Right? Okay, let's go ahead and stop there. All right, let me stop the recording.